Hello, everyone. Thank you for joining today's webinar. Today, we will be discussing how to reduce volume loss in fill finish for high-value drug products. The takeaways from today will include what types of volume losses should you expect and how much will you lose, techniques to reduce drug product loss during formulation and filling, risks of some techniques for reducing volume loss that should be considered, and an overview of the low-loss fill finish process at Sharp Sterile. Today, we have Vice President of Manufacturing Science and Technology, Dr. Shufeng Sun, and Vice President of Manufacturing, Tyler Rush. My name is Phoebe, and I will be your host for today. Very quickly, we welcome your questions during the presentation. To ask a question, select the bubble in the toolbar at the right-hand side of your screen. Write in your question and click Send. Now that that's out of the way, let's get started, shall we? First, I want to invite Tyler at Sharp Sterile to speak. Tyler, can you tell us what kinds of volume losses we should expect and how much will be lost?
Hi, Phoebe. Yes, I can. I will map out a typical formulation and filling procedure and outline where a product is lost during the process. Of course, the amount of volume that is lost will all depend on the project, the filling line, and the CMO. Many CMOs do not focus enough on reducing product loss, and their losses are often much higher. But for today, I will mention typical amounts that are lost on our filling lines at our site in Lee, Massachusetts. So let's start with formulation. Typically, we prepare the bulk drug product solution in a large vessel, usually a spinner flask. If the product is a simple thaw, pool, mix, and filter, then we may keep it in the container that it arrived in or transfer it to a spinner flask. So the drug product solution is prepared and we do not have any drug product loss. Next, we sterile filter the solution. For this, we have tubing that pulls the drug product from the spinner flask. The solution runs through two sterile filters, a bioburden reduction filter and a sterilizing filter. Then, this solution is transferred to a new pre-sterilized vessel. This can be another spinner flask or it could be a biobag. This process involves a lot of product loss. So let's list those. First, you will have product left over in the vessel you are pulling from. This will only be a small amount of loss, and it all depends on the volume of the drug product being prepared and the size of the spinner flask being used. However, for a 1 liter volume of drug product, you will likely lose around 3 milliliters of product. Line loss. This is a product that is lost in the tubing. The line loss during the sterile filtration is a significant part of the process, and the reason is due to the holdup of the volume of the sterile filters. Of course, you will have small droplets of solution remaining in the tubing after the transfer. At the end of the sterile filtration process, air replaces the space that the drug product solution was in. Once the end of the solution reaches the filters, however, the air cannot permeate the filter membranes. The pores in the filter hold small volumes of the solution, and the pressure required to dispel the solutions from these pores is very significant because of the surface tension that's holding the fluid there. As a result, the gas will not permeate the filters unless the system was pressurized, and even then, the gas permeating these filters would create a significant risk of rupturing the filter membranes and contaminating the drug product solution. So usually the gas is turned off before it pressurizes the filter past its bubble point. Once the gas does reach the filter, the solution will no longer move through the filtration assembly. Now the solution is stuck in the line after the two filters can usually be salvaged by physically massaging the tubing to push out the solution. However, the product that exists between the filters is stuck. This cannot physically be pushed out. The amount of volume that is lost between the filters depends on the length of the tubing, the inner diameter of the tubing, and the size of the filters. But if the diameter is a 1/4 inch and approximately 5 inches long, then you're looking to around 3 to 4 mils of product loss. A significant volume of solution will be stuck in the filters. This is called filter holdup and is generally the greatest source of product loss in either formulation or filling processes. The holdup of volumes will depend on the filter size, but for a 1 liter drug product solution, we can estimate that we will lose around 12 mils of product for each filter, so 24 mils of product for the two. We can estimate that about 5 mils of product is lost in the line as droplets or small volumes of solution. This is called line loss. This is all the volume loss that can be expected in a typical formulation and filter process. So our total product loss is 32 milliliters so far in the process. Let's discuss filling activities. At Sharp Sterile, the sterilized bulk drug product solution is connected to our isolator-based filling lines through a sterile aseptic connector, through a port in the isolator wall. The product is then pumped from the vessel through our filling assembly. The filling assembly is merely a long length of tubing that leads to our fill needle. Some of our fillers are double-headed fillers, in which case we have independent lines leading from a wide connector on the filling line within the isolator to each of the fill needles. The fill assembly needs to be filled with drug product and free of any bubbles or air that could cause inaccurate fill volumes. To do this, we pull solution through the tubing using a peristaltic pump, and we collect a predetermined volume of solution from each fill needle. This is called a line purge, and the volume that is typically collected is an arbitrary number based on what we believe is appropriate to ensure all the air is removed from the line. We can estimate about 10 mils will be collected for each filled needle during the line purge. If we only use a single head filling, then the loss will be 10 mils. But if we use double head filler, then the loss will be double, 20 mils. This is 10 to 20 milliliters of loss, depending on the line we fill on. Once the line purge is complete, we take 3 weight checks to ensure that we are filling within the correct volume range. After this, weight checks are only pulled every 50 to 150 units. Our lines, for the most part, use destructive weight check measures. So this loss is dependent on the volume of product that is filled into the container. But let's assume that we're filling 2 of our vials with 1 milliliter of drug product, and we're pulling weight checks every 100 units. Then, given all this math, you can expect to lose around 13 milliliters of drug product. This includes an ending weight check to bracket the remaining filled units. We also have line losses and solution remaining in the vessel we are filling from, likely around 5 to 10 mils. So, after filling is complete, we estimate that 10 to 20 mils is lost in line purges, 13 mils is lost in destructive weight checks, and 5 to 10 milliliters is lost in the line. This is a total of 28 to 43 mils of loss just in sterile filling. Combined with the formulation and sterile filtration, we're looking at 63 to 78 milliliters of drug product loss for this very specific example. This is about 6 to 8% loss in volume. As the volume of drug product is increased, the percent loss will drop because the line losses and filter holdup for much larger batches remains the same or similar to those smaller batches, unless the filters and tubing are sized up significantly. This will ultimately reduce the percent volume loss. There are other areas where API will be lost. Your CMO will strongly advise you to use API in the validation of your process. A CMO typically performs an engineering run or a mock GMP run to validate your process, and it typically involves formulating a full batch of drug product solution and filling a small portion of that batch, or all of it if the batch is small. The engineering run is usually non-sterile, and in no circumstances is it released for human use. These products can be used for research and development work, such as process characterization and analytical method quantification. Another area where product is lost is analytical testing. There are several required tests that must be done to each batch to demonstrate the product meets the set specifications and that it's sterile. Some of these tests are non-destructive, but most are not. Sterility testing, for example, is a destructive test and requires that it be performed for every batch that is filled. And this test alone uses 10 to 120 units. You will need to plan for these tests. So in review, here are all the areas in the process where drug product is lost and the expected volume loss for a 1-liter solution of non-viscous drug product. Of course, the numbers I listed in this example are only estimates, and the amount of product loss will be unique to each project, and they will be affected by the equipment that's used, container type, size of the dose volumes, and more.
Thanks, Tyler, for that process review. Now that we know where we're losing drug product, maybe we can answer how to reduce product loss. Shufeng, is that something you can answer?
Of course I can. Fortunately for our clients, there are several mitigation strategies to reduce volume loss. Let's discuss stereo filtration first. Filter holdup and line losses are the most significant source of product loss. There are several ways to limit this loss. First, you can reduce the length of tubing used in stereo filtration. This is the best mitigation strategy because it will cut down line losses significantly, and it does not add any additional stress on the filtration time and quality. You can also reduce the inner diameter of the tubing. This reduces the volume left behind the tubing at the end of filtration. But this will add stress to the system. If you continue to filter at the same flow rate, it may increase back pressure. But if you maintain pump rotary speed, it can reduce flow rate and extend filtration time. The tubing size should be optimized to reduce volume loss while minimizing the time spent filtering the product. If the product has shear sensitivity or hold time limitation, impact of tubing size changes should be carefully evaluated. You can use fewer filters. Filters retain a significant amount of solution, and use fewer filters will cut this loss. Using fewer filters will also mean that less solution will become trapped between filter membranes because there are fewer filter membranes in the system. The risk of using few filters is a potential reduction in sterility assurance when there's filter integrity issue. The potential filter integrity issue could be mitigated by performing pre-use post-sterilization integrity test. If you cannot use few filters, you may consider using smaller filters. Smaller filters retain less solution, but the size of the filter will affect filtration time. Smaller filters also filter less solution, so the filter size should be optimized for your process to reduce volume loss, but encourage an efficient and effective filtration process. One other volume loss reduction strategy is to filter into a bioprocessing bag instead of a glass vessel, such as a spinner flask. The reduction in volume loss won't be seen during filtration, but in filling. At the end of the fill, the biobag will be able to empty completely, while a significant amount of solution will remain in the spinner flask. Why? During filling, the dip tube must be fully submerged in solution. Once the level of solution drops, so the dip tube is not fully submerged, it will begin to siphon air. And this causes bubbles to become trapped in the fill assembly. Once bubbles approach the fill needle, filling activities are ended. Because bubbles create out of specification weight checks. Using a spinner flask during filling can sometimes mean 100 milliliter or more is left behind in the vessel. Biobag, because they drain from the bottom of the bag, will empty completely and are preferred when volume loss is a concern to the client. Now, there are two other product loss mitigation strategies that I will mention that have higher risk and should be implemented with extreme caution. The first is performing a filter blowdown. Solution trapped between filter membranes and in the filters is often the greatest source of product loss. A filter blowdown can be performed to achieve maximum product recovery. This is where stereo air or nitrogen is pushed through the filter and the filtration assembly to force out as much solution as possible. While this is effective at saving solution, it has many risks. Blowdown introduces additional manipulations and gases to the process, which may pose sterility assurance risk. Excessive pressure may stress filter membrane and cause damage to receiving containers such as biobag, which cannot withstand pressure. Foaming may occur at the gas and the liquid interface, which could be detrimental to some product. Because of these risks, we do not recommend this approach. If you really have to adopt a blowdown strategy to minimize product loss, the stereo gas pressure should be well controlled and maintained at the filter's bubble point. Connections to stereo gas are added before each filter for blowdown. It is also important to consider regulatory risk and ensure that the blowdown parameters validated in filter validation. Another high-risk strategy to reduce product loss is to filter on the line, which I and Tyler have described as our process for filtration and filling is two separate steps. First, stereo filtering the drug product solution into a second stereo vessel, and then connecting that vessel to a filling line to begin stereo filling. You can connect the non-stereo bulk drug product to the filling line and perform stereo filtration on the line. This means filtration and filling activities are taking place at the same time, and it removes the need for a second vessel or biobag to filter into and fill from. This also reduces the amount of tubing needed. However, the gains from filtering on the line come with a few risks. The filtration time is longer, which increases risk of contamination, product degradation, and filter clogging. You can only test the stereo filters after stereo filling is complete. If these filters do not pass, then all that drug product that was filled is going to be rejected. By splitting the stereo filtration and filling into two steps, we can test the stereo filters before stereo filling occurs. And if they fail, then we are able to refilter the product with new filters. This is to say that by filtering on the line, you risk losing your entire lot of drug product if the stereo filters do not pass integrity test. These are all the ways that the product loss can be mitigated in filtration. Next, let's talk about the filling. There are a few simple measures right off the bat. Reduce line purge volumes. The purpose of the line purge is to fill the fill line with product and to ensure that it is free from any air bubbles that could lead to inconsistent and out-of-spec weight checks. Air in the lines causes out-of-spec weight checks because the dispensed volume is calculated by the number of pump rotations. So if air is dispelled instead of drug product, the volume in that unit will be low. We perform line purges by collecting a large sample of the solution, usually about 10 to 20 milliliter from each fill needle. This number is generally recommended for minimum purge volume. That is usually high enough that we can ensure the line is free from air. For filling lines with reduced tubing length and size, you can set the line purge requirements to be less than what is typical to save drug product, and it will not affect your fill. The manufacturing team will be aware that they will need to watch the fill line for bubbles and ensure everything they see makes its way out of the line before they begin filling. So they may go over these limits if necessary. Otherwise, they will purge to the lowest volume, the purge requirement. Another way to save product is to reduce the tubing length and inner diameter. Again, tubing length will not affect your fill, but reducing the inner diameter will put more stress on the system, and it will require slower filling. So the tubing should be cut at the shortest possible length, and the inner diameter should be optimized to reduce volume loss without adversely affecting your fill. Weight checks. Let's talk about these. There are non-destructive and destructive weight checks. If your CMO is capable of non-destructive weight checks, then these should be performed. A typical non-destructive weight check is taking the weight of the empty container, filling it, and then taking the weight of the filled container. This is usually done in assembly line like fillers, where the containers get a weight while in line. It's also possible, but less common, on XY fillers. For these, you will need a system, typically a robot, that can remove the container or containers from the nest, place it on a scale to get a reading of the empty container, then place them back into the nest for filling. The same container or containers will be removed from the nest again to be read and placed back into the nest before it exits filling. You can see on screen an example of this that the stereo line has created. There are two common methods for performing destructive weight checks. The first is removing a filled unit, taking the weight of it and dumping the solution, wiping it dry, and taking a second weight measurement of the empty container and components. The difference between the two weights is the weight check. You can also perform weight checks on the line. This is where each fill needle dispenses the set volume into a weigh cup. The mass of that dispensed volume is the weight check. The latter is much faster as it can be done on the line. And most CMOs require a passing weight checks before they can continue filling. If your CMO has the capability to perform non-destructive weight checks, then that is a great way to limit product loss. If not, then you may want to consider performing fewer destructive weight checks. The number of weight checks performed is arbitrary. They are typically done every 50 to 150 filled units, and they are most often performed every 100 units. If your CMO has good control over precision of the dispensed weights, and they are not trending up or down as filling progresses, then you should be able to ask for a reduced number of destructive weight checks, switching from a weight check every 100 units to a weight check every 150 units will save a few units. For example, it will save 3 filled units in a 1,000 unit fill batch. Another great way to reduce product loss, and likely the best way to cut down the loss, is to perform single fill needle filling. If your lot size is small enough, then performing single needle filling will save a lot of product, because you will need the tubing for only one fill needle, and only one fill purge will be necessary. This significantly cuts product loss. We have 3 filling lines, and they all offer single needle filling. Even our semi-automated and automated lines, which have double head fillers, can be adjusted for single needle filling. Now, one last and simple way to reduce drug product loss at the end of a fill is to lift the bulk drug product solution. When you are near the end of the fill and you are pouring from a bio bag, the surface level of the solution will be awfully close to the tubing that is leading out of the bag and to the fill needle. When the pump runs, it is possible that the air can be pulled into the tubing. If the bag is hanging low and the tubing that leads to the fill needle travels upwards, then the bubbles may become trapped in the line, and they will work their way to the fill needle, ending the fill prematurely. The fill is ended when air becomes trapped in the fill line, because bubbles in the line signifies that is the dispensed volume will be out of spec. If the bag is raised, then tubing that leads to the fill line will travel down. If air is pulled into the line, the bubbles will have a chance to rise back into the bag while the pump is resting, as the XY table is shifting to place the next container under the fill needle. This prolongs a fill and allows the manufacturing team to completely empty a bio bag during filling. That is nearly everything that can be done in filling. So let's talk about other ways to mitigate the loss. Engineering runs. No matter which CMO you go to, they will perform an engineering run to verify your process. This is a mock GMP fill where the full batch of your formulation is prepared and a subset of that is filled. This ensures that your process works as expected before you begin manufacturing GMP lots that will be administered to patients. If your API is exceedingly expensive or difficult to procure, and if it does not have any unique formulation or filling challenges, such as suspensions, products that are shear or temperature sensitive, or life-raised products, then we may be able to use an inexpensive substitute instead of your drug product. This substitute will be prepared to have similar physical properties as your drug product solution. While it will not provide the same level of information as using your API, it can offer you the ability to perform an engineering run without using your high-value drug substance. Finally, let's discuss analytical testing. Planning out your analytical testing can reduce the overall number of units required for testing. One unit can be used to perform multiple tests. For example, if appearance and pH are required, then appearance can be assessed before pH is measured. You may also be able to opt for non-destructive tests to minimize the number of units required for analytical testing. For example, container closure integrity testing, or CCIT, can be performed by vacuum decay, a non-destructive test, instead of by dye ingress that is destructive. You should also discuss with your CMO these possibilities when you plan your analytical testing. So overall, these are the ways that you can reduce product loss. Reduce the length or inner diameter of tubing. Use fewer or smaller filters. Fill from a bioprocessing bag instead of mixing vessels or other hard material containers. Reduce line purge requirements. Perform non-destructive weight checks or perform fewer destructive weight checks. Use a single needle for filling. Lift the bulk drug product at the end of fill. Plan out your analytical testing to reduce consumed units. And use a substitute when this makes sense in engineering runs. Two techniques for reducing product loss that have higher risk than the above techniques are filtering on the line and performing a filter blowdown. These strategies, while they work at reducing product loss, should be the last resort due to the risk. In general, the risk outweighs the reward, in my opinion. I mentioned nearly all the ways that we can reduce product loss, but we still have one left to discuss. This last method is Sharp Stereo's low-loss fill process. 2 years ago, in one client's engineering run, around 290 milliliter of drug product was lost from a starting batch size of 310 milliliter. When we mapped out where this loss came from, about 180 milliliter was filled, but the units went to quality control and release testing, which including a sterility test validation procedure. This is a one-time test that eats up a significant amount of filled drug product, and 20 milliliter was lost to in-process weight checks. That left 90 milliliter of loss from the line losses, the line purge, filter holdup, and other areas in the process where the product will inherently be lost. While this is pretty good for the industry, the client was not happy because there were not enough units for clinical needs. So we worked together as a group with the formulation team, filling team, manufacturing, and validation to develop an entirely new process that was focused on reducing drug product loss. We decided to cut out nearly all the tubing we use. So instead of having several feet of tubing that connects our stereo bulk drug product to the filling line, we only have 3 inches of tubing. We will still have tubing for our stereo filtration process, but we cut it to lengths as short as possible. The product is stereo filtered into a pressure vessel. We currently have 2 pressure vessel options, a 600 milliliter vessel and a 1,100 milliliter vessel. Everything that is used in this fill process is single use. The product is collected in the pressure vessel. And then we use stereo gas at a pressure of 1 psi, which is very low pressure, to push the solution out. We use a dispensing valve that is simply opening and closing and opening to the needle, like a door. To increase the dose, we simply increase the time that this valve is open. We use an actuator to control the time that the valve is open, and the action is triggered by a foot pedal. During manufacturing, the operator will place a stereo vial underneath the needle, and they will lift the vial into place using a scissor lift that we have designed. The model you are viewing is a 3D printed model of this lift. The vial is lifted to the needle to prevent any risk of splashing, but the solution is moving slow enough that the splash should not be an issue. The operator presses the foot pedal, which triggers the valve to open, and the dose is dispensed. The vial is then manually stopped and removed for capping. There are only two factors that need to be tightly controlled: the pressure of the gas and the time that the valve is open. We must also consider that gas compresses differently than solution. So as the pressure vessel empties and the space is replaced with gas, the pressure may differ. In our studies, however, we found that both the pressure and the time were well controlled, and our weight checks remained within 1% of the set dose. This was demonstrated at doses even as low as 75 micrograms. Overall, between filtration and filling, we can complete fills with less than 30 milliliters of volume loss. Most of this comes from line losses, from filtration and filter holdups. Since we only have 3 inches of tubing to be concerned with air become trapped in, we can significantly reduce our line purge and our tailings. Tailings is the product left in the line at the end of a fill. In addition, we were able to perform non-destructive weight checks, which further reduces product loss. So let's talk about some advantages and the limitations of this filling process. The advantages include very little volume loss. There is significantly less product loss than a traditional filling process. This process also involves very little training. Only hand pipetting could offer less line loss. FDA and the other auditors do not like hand pipetting as a fill process because it is heavily dependent on operator training and has high risk on product contamination. The process we designed only involves loading a vial and pressing a foot pedal. The dosage and the repeatability is not influenced by operator training. The process is extremely accurate and very repeatable. This is ideal for shear-sensitive products. There's no mechanical pressing or pushing like you have with traditional filling processes, ones that use a peristaltic or piston pump to move the solution. And the system is under very slight pressure. It fills relatively quickly. For manual filling, we can likely reach a fill rate of 100 to 200 units per hour. Now, I should mention some limitations. Currently, this only works for vial filling. We may eventually be able to tweak the process to be able to process syringes or cartridges, but currently this is only designed for vial filling. And we cannot seal with a nitrogen headspace for oxygen-sensitive products. While we could push the drug product out with stereo nitrogen, the vial is still open to the air in the isolator. The headspace in the vial is not being pumped out of the vial and replaced with nitrogen. We may be able to find a solution for this eventually, but currently this does not serve as a solution for oxygen-sensitive products. The standard vessels are stainless steel and have a maximum volume limit of 1,100 milliliter. For product with larger batch size or with compatibility issue with stainless steel, we have developed alternative options using plastic or glass containers. However, some extra length of tubing is required in these systems. Although the loss is still significantly less than typical filling, it is not as big as the stainless steel option. With this fill line, we have been able to fill batches of less than 1 liter of product with fewer than 30 milliliter of product loss between line losses, filter holdup, and transfer loss.
Thank you, Shufeng. That is a very creative solution that you just shared. We've reached the end of today's presentation. In just a moment, we'll transition to the Q&A portion of the webinar. I'll give everyone a moment to answer their questions. Okay, it looks like we've received all the questions that we're going to get. Let's start with the first question. What is typical product loss for other CMOs?
The typical line loss at other CMOs, Phoebe, is really going to be dependent on that CMO's equipment, both for formulation and filling. But here at SSM, we've heard enough stories from clients that have worked at other CMOs to know that this is extremely important attribute and we must pay attention to. All of the operators and manufacturing are aware of this fact. From the engineers writing the batch records to the operators performing the operation, everyone's focused on minimizing the line loss and product waste.
Here's our next question. Does the amount of product loss differ for each filling line at Sharp Sterile?
The amount of line loss across all of our filling lines does not differ significantly. However, with the low loss filling line, we focus design of that line to minimize the product loss even more. This is designed specifically for clients that have very, very small bulk formulation volumes.
And the next question. What % of drug product solution should I be prepared to lose?
With the typical process and procedures for a 1 liter batch, you should expect to lose between 6 and 9% of your drug product. With the low loss volume filler that we're working on for the same batch size, we believe that we could be closer to 3 or 4%.
Another question. How many units are lost to QC testing?
The number of units that are required for QC testing really varies depending upon the product and the number of tests that need to be performed. Certainly, sterility and endotoxin testing need to be performed. Sterility is generally 2% or 20 units, whichever is less. Endotoxin is often just one sample, or it may be one from beginning, middle, and end. Then it depends upon what sort of analytical testing one requires. So it's very difficult to answer that. It varies from batch to batch. The macro testing is pretty well defined, but the other testing varies from product to product.
Just a few more questions. What is the smallest volume fill that you have completed?
The smallest volume that we have filled into container is approximately 200 microliters. But we can also talk about the smallest volume of drug product total that was filled. And that has been as low as 50 to 100 milliliter.
And our last question is, what is preventing you from using the low-loss fill process on all the filling lines if it has less product loss?
This process was designed for accuracy, repeatability, and precision. And to achieve those three quality parameters, we had to give up speed. So it really wouldn't be appropriate for large quantity fills.
Great. We'll end on that question. I want to give a huge thank you to everyone who attended this webinar, and a thank you to our speakers, Tyler Rush and Dr. Shufeng Sun. To all of our viewers that were here, keep an eye out for an email from Sharp Sterile after the presentation, and feel free to check out our website to take a look at other expert content from Sharp. I hope you took away a lot from this presentation, and I hope to see you at future webinars with Sharp.